Welcome back, everyone. We are back talking about that second level of biochemical structure, that of the lipids. Ultimately, where we left off last time, we had started talking about the basis of lipids, or fats, if you like, and the very basic building block of lipids are the fatty acids, saturated fatty acids, unsaturated fatty acids, and we were able to especially look at the unsaturated fatty acids with regards to some structural features. Of course, in these unsaturated fatty acids, we have these double bonds, at least one carbon-carbon double bond, at least one, and they all happen to be of the cis variety in nature, and that was essentially the difference between saturated and unsaturated fatty acids. We'll be looking more at unsaturated fatty acids because they have a diverse biological function profile. In particular, what we were looking at were essential fatty acids. All essential fatty acids are unsaturated, but in particular, we were looking down here on the bottom here, the omega-3 and omega-6 fatty acids as representative essential fatty acids. Really, all essential fatty acids are omega-3 and omega-6, and we had learned how to count in from the last carbon, the last methyl carbon on the unsaturated fatty acids, and it depended on where we encountered that first carbon of the multiple bond, either 3 carbons in or 6 carbons in, giving this omega-3 or omega-6 moniker respectively. Now, there is one particular unsaturated fatty acid. It is an essential fatty acid that I want to look at, and that was arachidonic acid. Now, arachidonic acid was one that we looked at. It happens to have 4 unsaturations within it at various points within there. I would never have you draw arachidonic acid from memory, but this one is going to hold a heightened importance because it is the precursor for many important hormones, 3 of which we're going to take a look at right now. Arachidonic acid, typically the way these fatty acids are put across are in one sort of straight line. However, for the purpose of introducing these new biological structures that are based upon arachidonic acid, I want to look at arachidonic acid written in a suggestive way. So over here, where it's sort of looped around upon itself. And the reason for that is that all of these types of structures I'm going to be showing you that are based on arachidonic acid as their starting material have that loop in there. So we'll be able to see the echo of that original carboxylic acid or unsaturated fatty acid. These structures are going to be known as the eicosanoids. There's 3 types of structures called the eicosanoids. Now, the reason for that, for the name anyways, is in Greek, the word eiko means 20. So you'll see that arachidonic acid has 20 carbons within it, and all of the eicosanoids, these hormones that are derived from arachidonic acid, have 20 carbons in there as well. So that's where the term the eicosanoids comes from. So let's look at these 3 different types of structures individually. The first one that has arachidonic acid as a structural precursor are known as the prostaglandins. The prostaglandins, notice it is plural because there are many types of prostaglandins within the lexicon of this particular hormone. It has a specific structure that one can recognize in relation to other types of structures. Again, you will be responsible for being able to recognize a prostaglandin, however, not draw it from memory on a blank piece of paper, say, or something like that. Now, when we look at the prostaglandins, structural features that just jump right out are this 5-membered ring, sort of this head group right here with various levels of oxygenation on it, and these 2 sort of tadpole tails, if you will, streaming off of it. With, again, again, different levels of oxygenation within there. And look over here on the upper right-hand side of every prostaglandin is that carboxyl group. That carboxyl group is echoed in the original arachidonic acid. So we see if we follow arachidonic acid around again and making this sort of loop right here, we can sort of see that echo again in the prostaglandin as well. It's obviously a different compound, but we see that that echo within there. Now, some important biological functions, there's a lot of them for the prostaglandins because, again, there are many different structures or different variants, if you will, structural variants of prostaglandins, and they all have different types of biological functions, 3 of which I've shown here. Prostaglandins are implicated in the inflammatory response within animal systems. They regulate inflammation. They're also a very different type of a type of biological use. They're implicated in gastrointestinal health. Gastrointestinal health, also in reproductive health. Certain prostaglandins are hormones within the reproductive system as well. So a vast array of different biological functions within these prostaglandins. The second eicosanoid we're going to be looking at, that hormone derived from arachidonic acid, are known collectively again as the thromboxanes. The thromboxanes, notice that is plural. There are a number of different structural variants of the thromboxanes. And again, just as we can see in that sort of curve around in arachidonic acid with our carboxylate right here, well, we see the same thing, that echo of that original arachidonic acid in the thromboxane with that, again, carboxyl group right there. So we see these, we can see how that they are related to one another structurally, even though they look a little different. With the thromboxanes, even though there are multiple structural variants of them, all of them, and this is different from how they're put across in prostaglandins, they're all only implicated in the blood coagulation cascade. They are essential, different types of, within the signaling pathway, different types of them regulate blood coagulation. It's all, the thromboxanes are all about blood coagulation. Now, if we look at the structure of this is the thromboxanes are similar in structure to the prostaglandins. However, if it has a head group. That head group we see right here in both of them, and it also has these 2 like little tadpole tails coming out as well. We can see a structural difference, however, in the thromboxanes. Whereas the head group on any prostaglandin is a 5-membered ring, the head group on a thromboxane is a 6-membered ring. It's actually a 4-6 sort of fused system with these oxygens within there, but it is structurally different. So if I showed you a listing with a prostaglandin and a thromboxane, could you pick one out from the other? Which one is the thromboxane? Which one is the prostaglandin? And so on and so forth. With fairly quickly, we'd be able to do that just based upon these structural differences. The last hormone derived from arachidonic acid, the last eicosanoid, are a series of structures known as the leukotrienes. The leukotrienes are the least structurally augmented, if you will, from the original arachidonic acid. There is no sort of head group on the end, such as we see on the thromboxanes or the prostaglandins, but we do so it's much easier to see that echo, if you will, of the original arachidonic acid. Here is that carboxyl group, which we again see on that arachidonic acid, and again we follow it around. We follow it around just like we did on the arachidonic acid, written suggestively there, so you can see the structural genesis of all of these eicosanoids from that original arachidonic acid precursor. Similar to thromboxanes, leukotrienes, even though that this is a plural right here, there are many structural variants of the leukotrienes. All of them are implicated only in the bronchial constriction cascade, the biological process. They regulate bronchial constriction, and there's only one purpose for them. Another type of general structure that is based loosely on lipids are the steroids. Remember in the beginning of this unit of study that says steroids were one of the three types of structures we were going to be looking at. Steroids, or whereas perhaps the eicosanoids or arachidonic acid are very easy to see that fatty acid precursor from them. It's based on fatty acids. Now, steroids are also ultimately based on fatty acids, but they're very far downstream in the constructive process or in the building process. It's very difficult, if not impossible, to see the original the original fatty acid framework in a steroid skeleton. So we'll talk about that a little bit more in a moment. But for some salient features on steroids, as I said, they're ultimately synthesized from lipid precursors. They have some carbohydrate within them. We're not going to go into the biological process of creation of steroids, other than to say they are a very downstream type of synthesis structure from fatty acids, so much so that we cannot see the fatty acid precursors. Now, steroids themselves are very easily picked out from a lineup. They contain what is considered the steroid carbon skeleton. That steroid carbon skeleton is a collection of 4 rings, and they are fused together. A fused carbon ring system ultimately means that they are connected to one another by at least 2 carbons. So here on this ring system, these 4 rings are fused together in the red that I'm putting out here. That's considered a fused ring system. There are four rings, and they are all given as particular letter A, B, C, and D. We always see this, and within there, with these rings, it is a 6, 6, 6, 5 fused ring system written like that. Once you get used to the structure of steroids, they're very easily pulled out from a representative lineup. But we find that they're very, very structurally different from any other type of lipid structure we've seen before. Over here on the right-hand side is another way of drawing this steroid nucleus, if you will. All steroids have this 6665 ABCD ring or fused ring system. It's just that they have some other carbon shrubbery upon them, slightly differentiating them from other types of steroid structures, but it's always very easy to point that out and say that is a steroid, because I see that representative in that very diagnostic fused ring system. So we'll be looking at, ultimately we're going to be looking at 3 important steroid structures and their biosynthesis of them. Now, we said that steroids are derived from lipid precursor. That lipid precursor is based on a structure known as squalene here. Again, even in squalene, which we see this sort of meandering carbon structure with some branching on it, we do not really see the echo of the fatty acid precursor within it, but we're not really going to go back that far. We're just going to look at 3 representative types of steroid structure that is based upon this squalene precursor. The first is going to be we're going to be looking at is known as cholesterol. Cholesterol, most of us have heard that term. It is based upon this steroid ring system, which we see here. And within this steroid ring system, we see various levels of, here's some oxygenation right here, here's some methyl groups, here's a whole bunch of carbon structure over in that sense. But it does maintain that original steroid ring system. So cholesterol is an absolutely necessary molecule within animal systems. It is found in cell membranes of most animal cells. It helps to regulate the fluidity of that membrane. We'll be looking at that towards the end of the this unit of study. Cholesterol tends to have a bad rap, if you will. It is implicated or high blood cholesterol cholesterol is implicated in heart disease and other health problems. As we can see over here from squalene going to cholesterol, our animal systems rather do indeed synthesize their own cholesterol, so it is indeed needed, and we maintain some cholesterol within our diets. So we have this pool of cholesterol in there, which is absolutely necessary for regulating fluidity within cellular membranes. However, it is only when we get too much cholesterol within our animal systems, within our biological processes, that we get these problems occurring, that of heart disease, other health problems, and that sort of thing. And it's just in the many times within the regulatory process of this cholesterol, if it starts to build up too much, we have to be able to regulate it and down-regulate it to have a particular concentration of it within our bodies. It's only when that concentration gets too high that we run into these health problems. But it's still absolutely a necessary biochemical structure within animal systems. The next type of steroid structure we're going to look at, and I should mention there, hundreds of different functions and types of steroid structures within animal systems. We're going to be looking at 3 of them. The second one are known as our bile salts. Our bile salts help to emulsify dietary fats in the digestive tract. So when you eat a fatty meal or something, what the bile salts will do is to emulsify those globs of fat, I know that's very appetizing, within our stomach, so they can be small enough so they're able to be uptaken by our intestines and brought into our bloodstream and towards our cells, so that we're able to utilize these fats. If we didn't have them, these globs of fat within our stomach and our digestive system would just go out the other end. Bile salts, there's 2 of them. They are synthesized in the liver and stored in the gallbladder. So when they are necessary, when they're called up, the gallbladder punches some bile salts into the digestive tract, and it helps to emulsify the fats in our diet or our digestive tract. There are 2 bile salts. They're fairly similar. There's cholate and chenodeoxycholate. On both of these, we see various levels again of oxygenation. Various levels of extra carbon structure in there. There's even a carboxylate as a cherry on top. And we can see that structurally it is different from cholesterol. However, and they are indeed different from one another. But there are slight structural differences that do separate them from one another. So both of these are excreted into the digestive tract when needed. The last type of important steroid structure that we're going to be looking at are the human sex hormones. Human sex hormones, the male sex hormone, that is testosterone, the female sex hormone, that is estrogen. Now, both of them, interestingly, are of steroid structure. Within the development of male and female sex characteristics, they are a very important signaling molecules. Interestingly, we hear a lot about both types, testosterone, estrogen, that sort of thing. It is interesting that they are both of steroid type structure. And they both have the function of regulating the development of male and female sex characteristics. I'd like to swing back and talk about one of our original functional groups, that of the ester. And I call them traditional esters right here. Traditional because esters are traditionally brought about by the condensation of 1 carboxylic acid and 1 alcohol, and we have our ester structure. If we look at our basic ester structure down here, we have what is shown there. That is what we have in the middle is sort of our general ester structure, but it's flanked on both sides by these R groups. That is considered hydrocarbon type carbon, and we have our traditional ester. Now, how this is based upon lipids is the basis of lipid structure, or rather the building blocks of lipids are fatty acids. On an ester, we have a portion of it that is derived from a fatty acid structure, or rather a carboxylic acid structure. So if that's brought about by a fatty acid, then we can say that certain esters can be derived from lipid precursor. So let's if we have a situation in which we have a lipid precursor, which would mean our R groups here are long chain. We have long chain R groups on either end, or long carbon chains, such that we would have an ester bow in the middle, if you will, and extending out from that, these R groups would be very, very, very long. So when we have that sort of situation, we run into, or we define what are known as natural waxes. Waxes are long-chain traditional esters. A wax, we can think of candle wax, or we're all familiar with what waxes ultimately are. But if we had to put a definition to it, ultimately waxes are esters. Waxes are esters, but they're derived from the condensation, yes, carboxylic acids and alcohols, but both sides are very long chain. Waxes, as we know, are typically solid at room temperature, and they're very hydrophobic due to the high hydrocarbon content within there, little or no polarity, no hydrogen bonding. Hydrophobic means waxes do not dissolve in water. Hydrophobic, or they don't like to associate with water. So important here is that the waxes we're talking about are natural waxes. They're biological waxes, and we can look. There's many waxes out there. We'll look at a couple of them. The first example, a good example of a natural wax, would be beeswax. Now, beeswax is made up of many different compounds, and it is a malleable natural wax derived from the thoracial glands of immature honeybees, to put a term to it. Essentially, honeybees make their own housing. They make this wax because they extrude it, and they'll literally build their own house with these biological extrusions from their own bodies. Now, as I said before, beeswax has a number of different compounds within it. Chiefly among them, however, is this particular ester right here. Look at this. This is indeed an ester because we see that ester bow right in the center there. But on either end, on the carboxylic acid end, we see a very long carbon chain, indeed 14 carbons. And on the alcohol portion, we see a very long carbon chain, indeed 26 carbons. So that put together, if we were to isolate this particular compound, it would have the attributes of a wax. Another example of a natural wax would be called that of carnauba wax. It's a very, very hard natural wax derived from the leaves of the carnauba palm. Look at this one right here. Here is our ester, and there's the carboxylic acid portion, 26 carbons. Here is the alcohol portion with 30 carbons. And within it, this is a much larger, if you will, than the beeswax we saw before. While beeswax is fairly malleable, carnauba wax, because it's larger, is very hard. Typical uses of carnauba wax are waxing fine furniture or fine woods and that sort of thing. Now we're going to move into what I'm considering non-traditional esters. Remember, a traditional ester was derived from one carboxylic acid condensing with one alcohol. I consider that traditional. Moving into non-traditional esters now, we're talking about what is known as glycerides. Glycerides are long chain because they're derived from fatty acids, but they're non-traditional esters. They aren't derived from one carboxylic acid, and 1 alcohol. Indeed, we're going to start our discussion of glycerides with what are called neutral glycerides. Neutral glycerides essentially means that they are neutral when we look at them, and that should, for many molecules, should go without saying. They don't have charges on them, positive charges or negative charges or that sort of thing. It should be obvious, but we're going to look at some charged glycerides that down the road that differentiate themselves from neutral glycerides. So that's why we term these neutral glycerides. As a definition, neutral glycerides are lipid esters, as we saw before. Lipid, derived from long-chain fatty acids, derived from the condensation of a special type of alcohol, and that is known as glycerol. Glycerol is a very important biochemical structure, or biological structure, in which we have 3 carbons, and on each of those carbons we have a hydroxyl group. So it's like a tri-alcohol, So glycerol has 3 hydroxyls on it, and if one of these hydroxyls happens to condense with a carboxylic acid, how about the carboxyl group on a long-chain fatty acid right here? What can happen once it spits out water is that we get an ester linkage. So between this hydroxyl and this carboxylic acid, we can, on one of the hydroxyls of glycerol, get this ester linkage that we see right here. So when we have one fatty acid connected to one of the hydroxyl groups on our glycerol molecule, we have what is known as a monoglyceride. Now, in this type of structure that we see here, it does not matter the specific identity of the fatty acid. In this case, I have a fatty acid that's a saturated fatty acid. It is a certain number of carbons long. It does not have to be this particular molecule. This is just being used as an example. We have a representative, if you will, fatty acid condensing with one of the alcohol groups on glycerol, and we get a representative monoglyceride. How do I know it's a monoglyceride? Because on the 3 alcohol groups on glycerol, only one of them is tied up in an ester. So therefore, this would be a representative monoglyceride. Now, of course, we have 2 other open hydroxyl groups on our glycerol, and if those are connected to long-chain fatty acids, we can have 2 other types of structures. We can have diglycerides, in which we have 2 of our alcohol groups on glycerol esterified, if you will, with a fatty acid. We have 2 ester groups. Again, they have to be long-chain fatty acids. We would have a diglyceride. Notice, again, in this diglyceride, I'm using representative molecules. This one on top here happens to be a saturated fatty acid of a particular carbon number. Doesn't have to be saturated, doesn't have to be that particular carbon number. Notice the second fatty acid happens to be an unsaturated fatty acid. Doesn't have to be this particular one. It's just going to show you some representative fatty acids that are condensing with glycerol to give, in both of these cases, we have a monoglyceride and a diglyceride. Should this happen a third time, we have a triglyceride. All 3 of our alcohols on glycerol have been condensed with a fatty acid. They're different fatty acids here. Again, it doesn't matter which ones they are, but this would be a representative triglyceride. We're going to find that triglycerides have a heightened degree of importance in energy storage. Triglycerides, when we think of fats as an energy storage system, indeed, fat tissue are basically storage of triglycerides. Triglycerides as a store of energy are stored in particular modules within animal systems. So here we have what are known as adipose tissue, or rather adipose cells right here. These are essentially fat cells. And fat cells are basically large cells with most of their functionality contained around the periphery within the cell. We have the cellular nucleus, we have the various cell organelles, but the vast majority within these fat cells are storage containers, and these storage containers contain triglycerides. So these triglycerides are held within a module within fat tissue until such a time as they are required, and then they are broken down. The triglycerides are broken down into fatty acids, and they are going to be catabolized for the structure, for the energy within them. Moving from neutral glycerides that we just looked at, mainly the triglyceride, which is a store of energy, I had told you we were going to start looking at something called charged glycerides. And to do this, we have to rethink our idea of the state of certain functional groups when we think of organic chemistry versus biochemistry. In organic chemistry, we had functional groups, or carboxylic acid, say, or amine, or even a phosphoester. Remember, our phosphoesters were a carboxylic acid derivative we talked about. And we think of these, when we think of them in a test tube, we just think of them written as they normally are. There is no convention for different conditions. Whatever the conditions we have are those within the test tube. It doesn't matter. However, in biological systems, things are different. In biological systems, typically animal systems, we have within, whether we're talking about within the cell, within extracellular mass, within digestive systems, and so on, we have particular conditions. Notably among them are the pH, which is terming as physiological pH. Physiological pH, at least within the bloodstream, is pH 7.4. 7.4. And this is exquisitely maintained. It's always pH 7.4. Now, there's certain functional groups which are ionizable at that particular physiological pH of 7.4, three of which, very important points, three of which are shown right here: the carboxylic acids, the amines, and the phosphoesters. So let's look what happens when we place a carboxylic acid, an amine, or a phosphoester into an environment that is exquisitely balanced to have a pH of 7.4. Up on the top, let's look at what happens to the carboxylic acid. The carboxylic acid at physiological pH of 7.4, that physiological pH actually acts as a base. A base all around that carboxylic acid, such that the carboxylic acid always loses a hydrogen ion and exists as its carboxylate salt. Always at physiological pH. It exists as a carboxylate salt. Never again at physiological pH will a carboxylic acid exist as a carboxylic acid, as we see here. It's always as a carboxylate salt. Similarly, we saw that an amine was another ionizable functional group at physiological pH. Physiological pH with respect to an amine acts as an acidic system. So remember, when we treated an amine with a strong acid, what we ended up with was an alkyl ammonium salt. And in the case of any amine now at physiological pH will always exist as an alkyl ammonium salt. Notice the difference here. On the amine, that lone electron pair reaches out and picks up a hydrogen ion, so that's really what we're looking at right here, and we have an overall positive charge upon it, such that no longer do we ever have just an amine at physiological pH, when this happens to be a primary amine. It could be secondary amine, it could be a tertiary amine. They are always existing as alkyl ammonium salts at physiological pH. Lastly, Our phosphoester. We haven't talked a lot about reactions of phosphoesters and how they exist when treated with acids or bases. All we've done up until this point is to recognize this is indeed a phosphoester. We find at physiological pH that physiological pH acts similarly to that of a carboxylic acid. It acts as basic conditions, such that these two hydrogens on the phosphoester are actually acidic because it is ultimately derived from phosphoric acid, so they are acidic. Put into a situation, physiological pH acts as basic conditions. A phosphoester always exists as its dianion. Dianion exists as a 2- ion at physiological pH. On the ester portion, of course, is not ionizable. That just stays as it were. But on a typical phosphoester, which we're going to be seeing in these charged glycerides, always exists as a 2- anion. It is charged. These two negative charges at physiological pH. Having said the previous, let's take a look at what ties glycerides together. We'll be talking specifically about charged glycerides, but when we think back about neutral glycerides are tied together with the molecule of glycerol. That's that 3-carbon structure. On all 3 carbons, we have an alcohol group. So glycerol tethers together neutral glycerides that we looked at previously. Now, when we think about, we're going to start to introduce what are called charged glycerides. So we're thinking about structures at physiological pH. When we think of a charged, one of those ionizable groups we looked at before, the amine, the carboxylic acid, and especially the phosphoester, they are charged at physiological pH. In charged glycerides, we get glycerol, which has formed what is called a monophosphoester. So here's that glycerol portion right here, where that last alcohol on glycerol sort of turns around here. We have free rotation around it, but it is then esterified into a phosphoester with a phosphate group. So we do indeed have that phosphoester linkage. We can see that P double bond O right here, oxygen, carbon. That is a crucial linkage within charged glycerides. And indeed, all glycerol molecules are covalently bound to a phosphate group when put together, when we're going to be putting together these charged glycerides. So charged glycerides, again, are based on this glycerol. Excuse me here, glycerol attached to our phosphoester. So that's what we looked at in the previous slide. Now, what we want to look at now are the other 2 alcohol groups on glycerol. That would be this one, and they now are connected, or rather condensed with 2 fatty acids in an ester. So we have these 2 fatty acids now, which condense with the alcohols on the remaining on that glycerol to now form a basic structure. And if we look at this right here, it is the basis of a charged glyceride. It is a condensation of our neutral diglyceride. So here is our diglyceride, if you will, because there's 2 fatty acids that are covalently bound to it, and it is covalently bound to a phosphate group. So we're always going with a diglyceride connected to a phosphate group. Because we are now at physiological pH, we have that the two OHs on the phosphate group are charged. They are ionized at physiological pH. We have a double negative charge upon there. And this basic structure now is known as a charged glyceride. They have vastly different functions, charged glycerides do, than neutral glycerides. If you remember, our neutral glycerides, and particularly the triglyceride, is a form of energy storage. That's what it is. It's a form of energy storage. The charged glycerides are fusion of a diglyceride with a phosphate group, are the primary structure or building blocks of biological membranes. So the base structure of this is a molecule known as phosphatidate. Phosphatidate essentially is a diglyceride covalently bonded with a phosphate group. So we have what is called a phosphatidate structure as a base structure. And it is through this phosphate group right here where we'll have a covalent attachment of small molecules to give 3 basic building blocks of biological membranes. So let's tie all this together. I said we would look at 3 small molecules. Here they are. We have in front of us these, and I've drawn them in red to be somewhat suggestive when we look at the large structure. The first one is known as ethanolamine. Ethanolamine, we can think of this structure here as ethanol, and it has an amine attached to it, therefore ethanolamine. If we structurally augment ethanolamine, when we have over here we have an NH3+, if we took all 3 of those hydrogens and instead replace them with methyl groups to give something like this over here, leaving everything else the same, we would have a small molecule known as choline. So we have ethanolamine and choline thus far as our 2 small molecules which are going to covalently bind to our phosphatidate. Lastly, is actually one of our 20 amino acids, which we're going to look at when we talk about proteins, serine. Now notice all of these have a hydroxyl group right here, that all of them will be able to covalently bind to phosphatidate through that. Serine just happens to is one of the amino acids which happens to have a hydroxyl on it, just like choline, just like ethanolamine. So these are our 3 small molecules which we are going to covalently bind to phosphatidate to give our 3 major building blocks of biological membranes. And let's see where these 3 structures attach to on the phosphatidate overall structure. So if we covalently bind ethanolamine to phosphatidate, all of these small molecules are going to be attached through one of the negatively charged oxygens on the phosphate structure, throughout. And ultimately, we have a covalent attachment between, for instance, ethanolamine, which we have over here in the upper left-hand side here. There is ethanolamine, and there it is covalently bound to the phosphate on phosphatidate. So what we have here in the upper right-hand corner here is a contraction on that word, phosphatidyl ethanolamine. Phosphatidyl, that comes from phosphatidate, ethanolamine, because that's the small molecule that we're ultimately working with. Phosphatidyl ethanolamine. If instead we have our phosphatidate molecule covalently bonding with a molecule of choline through that same oxygen on the phosphate, we have a structure known as phosphatidylcholine. Phosphatidyl is the same prefix, if you will, on all 3 of these building blocks of biological membranes, but it is the last part, that suffix, that is different, that shows which of our small molecules is ultimately bound to it. Lastly, let's take a look at our serine molecule down here. Serine binds through its hydroxyl group with phosphatidate to give phosphatidylserine. Phosphatidylserine, and there's that connection point. As with all of the other ones, we now have 3 types of structures put together. We have our phosphatidyl ethanolamine, phosphatidylcholine, and phosphatidylserine. Now let's take a closer look at these structures. It is only on one particular area that we see charged structure on all of these. Let me circle that area, and we see charged structure all over the place. Up on top, phosphatidylethanolamine, we have a negative charge here on phosphate. We have a positive charge on that amine. Remember, at physiological pH, any amines are positively charged. Similar here on this amine on phosphatidylcholine, the next one down, we have a positive charge. We have a negative charge here remaining on the phosphate. On the last one down, on phosphatidylserine, we have a negative charge on the phosphate. A negative charge on this carboxylate on serine. Remember, carboxylic acids at physiological pH always exist as their carboxylates. And on the amine we see here is positively charged, ionized at physiological pH as well. So we have this area here that I've circled in blue of a charged area, and the area I'm circling in red now, we can see is an uncharged area. There's no charges whatsoever on here. So we're going to see that we use this conflicting type of structure. We have charged areas and uncharged areas on these building blocks that are going to allow these biological membranes to form in an aqueous environment down the road. We'll look at this in an aqueous environment, very similar to these the soap mice cells we saw forming when we were talking about the structure of soap.